It's strange enough, as you see on this slide, here is the most picturesque part of the St. Petersburg, known as a spit of the Vasilievsky Island. Probably you have already know that. This is the spit. Here is famous museum, former stock exchange. And here is our famous institute, just between stock exchange and university, the State University of St. Petersburg. And this there is a good, interesting history. The institute has more than maybe 100 years old. He was founded on the money given by the Nikolai II to obstetrician Ott. He was German by origin and living for many years in Russia. And using this money and also some donation from the public, they constructed this building in memory of, it was the gratitude of the Nikolai II to Ott for his taking the birth of Alexei, the Princess Alexei, which later was, as you know, killed. But at the beginning, it was like that one. And here is... На двух мне будет сложно. Я должен...
Да, прошу прощения, сложно понять, с какого языка на какой мы переводим, но это изображение Института акушерства и гинекологии имени Отто, у которого довольно интересная история. Он был основан на деньги, подаренные Николаем II известному немецкому по происхождению врачу Отту когда-то, и этому институту сейчас уже более 100 лет. Он находится в очень живописной части нашего города.
I'm afraid I should be more concise. Really, I need to be translated in Russian. Ну, так переводите.
Ближе микрофон держите еще раз.
Следующий слайд. I understood nothing, no words of the previous reporter, and understood how far we are in our scope of interest. Я понимаю, как мы далеки друг от друга, и поэтому I've decided to start my talk showing you the entity of human heredity. And this entity, as you know, is confined to the number of cells, great number of cells. I will tell you how much. Each cell has its so-called genome, which we'll speak about today. Genome is composed of 46 chromosomes, and there is now known as there are about 23,000. There is written 23,000 of genes, and the genes and this famous double helix of DNA, and the two strands are tied by the special blocks of nucleotide. And here they are written. I will not name them. And all this rather modest amount of genes are responsible for synthesis about 300 of different proteins. Смысл сводится к тому, что вы, наверное, поняли, 46 хромосом. В каждая хромосома представляет собой единую нить ДНК. Она может быть развернута. И каждая нить представляет собой соединение, собственно, двойной спирали, известную, знаменитую, и открытую в 1953 году. И геном человека содержит 23 тысячи гена, и они ответственны за синтез 300 тысяч белков. So this is a background for my following presentation. And here are the main stages of human genome studies as you see them on that slide. So here are Bill Clinton, known for each of us. This is Francis Collins, the chairman or president of Human Genome in the United States, now the chief of FDA organization of the United States. And he's Craig Venter, the private owner of very complex enterprise with which he succeeded to sequence genome earlier even than it was made by international group. Я немножко затянул здесь. Итак, Коллинз, Клинтон, Крейг Вентер, владелец частной фирмы, которая конкурировала с международной организацией секвенирования генома человека и сильно в этом преуспела. И, as you see here, and it shouldn't be translated into Russia, that this year, 1990, 2003, this is when the genome project was accomplished, fulfilled, and as the goal of this project was to sequence this very long, about 1.5 meters molecule of DNA which are represented in human genome. And this was the first stage. It cost roughly $3 billion at that time. Second period was that it was when the also international group of scientists, they made a mapping in genome, molecular marking, how they are distributed along chromosomes, along DNA, and how can they be used to pinpoint the genes inside the human genome. Значит, они картировали специальные ДНК-маркеры, так называемые СНПы, как они распределены по геному, чтобы к ним привязывать картированные гены. This year, 2007-10, this was the first individual genomes were obtained, were done, and the price for each genome was roughly about $10 million. But later, because of serious improvement in the sequencing technique, you see here already during the rather close to us, the current time, 2008 to 11, first there was genome of 1,000 genomes. So the genomes of 1,000 persons, mostly in Europe, they were sequenced. And they included 4 different populations for that, and these are the geographical regions from which this population were taken. And European genome as a whole, as a work, separate work, based on the sequences of 1,500 persons, was accomplished in 2013. Now in progress is very advanced English project, which relies on the sequencing of 200,000 men genome. And not only simple sequencing, but also correlation of medical chart of each person with peculiarity of its genome. This is rather important. Они не только секвенируют 100 тысяч геномов, они уже сделали примерно около 70 тысяч, но они сравнивают особенности генома каждого человека с особенностями его медицинской карты, с теми болезнями, какими он болел, и с его результатами лабораторных исследований. With some pride, I should say that at the end of earlier this year, better to say, we started so-called Russian genome. Nobody exactly knows what are Russians in general. Nobody knows what's a Russian genome, because it's difficult to say who should be collected to represent the Russian genome. Само по себе понятие русский геном довольно сложное, как трудно определить, что такое русские люди. Но, тем не менее, такая программа начата, и она ведется, и она предполагается, что будет использована примерно 3000 человек. At the end of 2015, there was astonishing news that at least succeeded to sequence all genome for 24 hours, and for the cost only $1,000. Without interpretation, frankly, without interpretation. Just dull sequencing the order of this nucleotide in this chain. Действительно, вот такая была сногсшибательное известие, что за 24 часа удается секвенировать геном человека, правда, без интерпретации результатов этого секвенирования. Ну и стоимость, если, так сказать, по минимуму брать, действительно могла быть всего $1000. Я чувствую, что я могу увлечься, поэтому я постараюсь довольно лаконично говорить, насколько мне удастся. I try to be as concise as possible. So the most advances in the area of human genome, where we are now in the studies, are shown on this slide. So first, the very efficient method for genome-wide association studies, so-called GWAS method, was introduced using this method over almost all, but the most important so-called multifactorial diseases. These are just common diseases. The genes for these common diseases were identified by this method. And for each disease, they succeeded to find so-called genetic profile, including the genes for aging also, so definite genetic profiles. What does it mean, genetic profiles? There are two profiles. One are what genes are involved, and the other is how they are working, these genes. So it can be functional profile based on RNA analysis, or it can be DNA analysis. То есть профили генетические, когда здесь написано, имеется в виду и наборы генов, которые вовлечены в ту или иную функцию, и их функции, или так называемые экспрессионные профили этих генов. A very peculiar and very complicated story, which will come a bit later in more details, concerns so-called missing inheritability. Suddenly now notice that the genes can't explain everything, all details. I will come later to that. But just to explain the action of the genes, one should know what is it epigenetic. These are external influences, the factors which stimulate the action of the gene, and thus they regulate the action of the genes. Я подробнее расскажу, это действительно очень важно, но мы вернемся просто на более поздних стадиях. Systemic genetic, this is to study metabolic pathway and the reaction of genes at different stages of development. Quite unusual but very important area now is so-called microbiome. Each of us has about 1.5 kilos bacterium in our intestine. Not so little. As I told you, the man has 23,000 of genes. A number of genes in this microbiome is several millions. So we have such a great population of different creatures living inside us and how they influence our behavior and our health. This is what they started. Я хотел сказать, что микробиом действительно привлекает сейчас очень много и в медицине, и где угодно, и поголовно занимается целой лабораторией на Западе. Речь идет о микробиоме, как те микроорганизмы, примерно 1,5 килограмма, которые живут в каждом из нас и которые влияют на наше поведение, питание, настроение и так далее. Mostly I will speak today about the predictive personalized medicine and the consequences of this initial predictive medicine, we so-called precision medicine in the United States and preventive OP of three-pair medicine in Europe. And the other discovery, very spectacular discoveries now, are the possibility to make correction inside the human genome by special techniques, so-called, he has written in Russian, редактирование генома is just, if you see some mistake, you can correct it. If you want to spoil in particular place, particular gene, you can also use this method. No, and as a last, quite a fantastic area is a synthesis of life. The synthesis of genome which provides the life processes. And this is already done for definite bacterium. So bacterium which do not exist, they are synthesized. And so, I don't know, some idea is so-called white project, which maybe not receive support to synthesize the human genome. From the very beginning to this functional, to synthesize human genome with all this I can't understand how it can be done, but this project was addressed this year, earlier this year. Удивительный совершенно проект, который, я думаю, к счастью нашему, и, может быть, человечество не будет иметь продолжения, искусственный геном человека. Ну, это к чему можно привести, это трудно представить. Думаю, что ни к чему хорошему. Пожалуйста. This is just briefly. Это известная молекула ДНК. And these are these very sophisticated machines which are used now for sequencing, for cutting this DNA into pieces. The idea of sequences is just to understand which nucleotide is followed or followed by another nucleotide. So the sequencing is to write the string of these nucleotides. So this is a rather weak ion torrent. We have it in our lab. We also have this sequence, and this is very expensive and very highly productive sequencing, high-sec, and so on. What we could expect from the sequences of the whole genome. First of all, we expect that new mutation associated with particular disease will be found. We will understand more details of how the copy number variation, there exists such a variability in human genome as copy number variation, how they influence these common diseases. We will know more about the peculiarity of human genome work in particular tissue. We will know more about the individual variability of genome and the genetic specificity of the disease. We know that each of us has its own, though the disease may have some common name, and you can remember a lot of these diseases, but each of these diseases for each of us is different, and because of peculiarity of our genome. This is what they want to study here, the biomarker of these diseases and models of biological models of this, or computer models of these diseases. Девушки. Это придется еще. I should now tell a few words about predictive medicine as a second part of my report. And as you see, to my opinion, the marriage or alliance between medicine and genome studies resulted quite naturally in production such interesting branch of medicine, like predictive medicine, and it appears in around 200 years at millennium and the eve of millennium, and already gave the start to many other kinds of genomic here there written. It's based mostly on the finding and testing of so-called predisposition genes. That not all the genes are involved in particular trait or particular disease, but some specific set of these genes. And these are called predictive genes. as we have, each of us has its own genome, so its study suggests that the medicine should be individual or personified, and it should have a prophylactic direction, because it's easy. You can take and study DNA at any stage. In my laboratory, we take DNA from the fetuses, and we can get knowledge of human genome before birth, and so the genome doesn't change much throughout the life. Predictive preventive personalized medicine give a chance to find inherited predisposition to common diseases long before their appearance. And the crucial point of predictive medicine is the idea of genetic path, or the set of the data specific for each particular person, which reflects the peculiarity of the genome, of each human genome, and how it's related to the health of the person. Пожалуйста. And this is some stages of evolution as I understand it. First of all, to my knowledge, it was really the first. When we edited the book, and it was really made in millennium age, it is called Human Genome and Gene of Predisposition Genes, Introduction into Predictive Medicine. This book appeared and is now rather rare. And in 2009, we edited another book which combined all our data, put them together. This is a genetic path as a background of individual predictive medicine. This all appeared from my laboratory. So here are the main data as I understand them. To be honest, I should say that in 2008, American scientist whose name is Leroy Hood, professor, who is now working in Seattle, he suggested that instead of, there should be not 3P as we call it, 3P medicine, but there should be 4P medicine, because the fourth, he thought this medicine should be participatory. It's rather difficult for translation in Russian, what does the word participatory means, but actually it means that the man should sам participate in his own treatment. Там довольно сложно перевести вот эту новацию, которую сделал Лерой Худ. Он сказал, что не только предиктивная, превентивная и персонализированная медицина, но она еще и participatory, или, я даже не затрудняюсь сказать, то есть participatory – это нечто подразумевающее, что человек сам принимает участие в создании и применении вот этой вот медицины. Вот что-то в этом духе. Ну, после этого была очень замечательная стадия, so-called translational, or so named targeted medicine, when we use our knowledge on metabolic pathway and know in what stage we should act and what substance we should take to correct the reaction. And in 2015, there was the era of precision medicine. We come to this later. And please pay attention that the main goal of precision medicine, as it is understood now, is the creation of so-called electronic genetic chart of the health. So we start simply from genetic chart or genetic path, as I called them, and we now are coming again and after several years, we are coming, after almost 20 years, we are coming to electronic part of this chart. This is the first original gene path as I understood at the start. And here are that first of all, that it is strictly confidential. We thought about that in 1997. What, besides genetic counseling, which is quite normal, what should we think it's worthwhile to know for your own, for the genome of everybody, is pre-symptomatic diagnosis of genetic diseases with late manifestation. Here they are reflected, I will not speak about them. Then the most important, especially for the spouse, are the knowledge of, aren't they the carrier of some abnormal genes which can cause troubles in their embryos, which can can result in the bone the child with inherited diseases. And for that reason, it's possible to make the diagnosis of couples for these particular diseases. But the couples are quite healthy, and they are simply heterozygous. And in heterozygous, many diseases are not manifested. And this is important because some of them are very often, like cystic fibrosis, for instance, hemophilia A, and so on. So, and really it's now in practice in many countries, in Israel, to my knowledge, and in Europe, for some of these diseases are tested very efficiently. And now on the right, there is also the diseases which we think is possible to predict if you give a testing of the gene, of the listed genes. Девочки, вы хотите, я буду нажимать то же самое. Мне не сложно это делать. Так, now I've told you about this, the mystery of missing inheritance or missing heritability, which struck us. It was very severe. I don't know, удар как будет, hit. It was really a very severe hit for me when we realized that. And this slide shows that genome is not responsible for everything. You can see the data taken from the analysis of the twins, different analysis. And you can see that when the twins are compared, the height is the genetic impact is only 80%, and 20% are coming from outside. Body weight, the color of eyes, shape of ears, and diabetes mellitus. This is the coincidence which shows that in diabetes, you see, not only if you are born, if there is a risk of schizophrenia, for instance, it doesn't mean that both twins will be affected. There are really a high chance, but not absolute chance, though they have the same genome. This is далеко не всегда, даже при риске рождения, скажем, ребенка с шизофренией, при наличии близнецов, оба близнеца болеют шизофренией. Точно так же это касается диабета и других заболеваний. Это только означает, что по-разному проявляются гены, есть какие-то факторы внешние, которые влияют на это проявление, которые бы надо знать для того, чтобы руководить этими делами. И отсюда так плавно мы переходим к довольно сложной проблеме, которая привела это missing inheritance, или отсутствующая наследуемость, когда стало очевидно нам, что мы можем понять, можем добавить наше познание генома тем, что мы будем исследовать его не как мертвую анатомическую систему, как орган какой-то, геном, а будем исследовать его в динамике, мы будем исследовать рабочий геном. А для того, чтобы исследовать, как работает геном, вот здесь показаны стадии работы этого генома. As you see, these are genes, information RNA, peptides, and this is the stream of information, if you wish. then a factor of regulation, metabolic pathway, functional models, and cell. So all they are involved. So if you want to understand the trait at the end, we should pass through all these stages. Only in that case, we'll gain more information about the working genome. Only in that case, we can approach for more precise prediction of multifactorial disease. This is what I will not take all your attention, or I will not misuse it. Не буду злоупотреблять вашим вниманием. Though I think it's important what basic results we got using this new approach in our studies. Я покажу на двух примерах, что мы, так сказать, смогли достичь, используя системный подход к генетике. Есть два серьезных заболевания, одни из наиболее серьезных и частых. Значит, так, примерно 40%, а с возрастом и больше женщин имеют миомы матки, и где-то порядка 10% имеют эндометриоз. То есть, по сути говоря, не найдешь ни одной здоровой женщины. И это действительно огромная проблема. Это проблема с деторождением, проблема с социальной адаптацией. В общем, мужчины могут быть в чем-то рады, что мы не подвержены таким испытаниям, каким подвержены женщины, имеющие эти заболевания. И эти заболевания являются ведущими в нашем институте, они изучаются в течение многих лет. И we made two types of experiments. First with endometriosis, and this data appeared in European Journal of Obstetric Gynecology and Reproductive Biology at the end of the previous year. And as you see, the basic conclusion of this work is that here you see a feasible way how these cells from which give rise to endometriosis, there are different routes how these cells can be formed, and later their development is also not unanimous. They can deviate in different channels, so-called epigenetic landscape, which depends on genetic loads of particular person, of his hormonal impact or peculiarity of action of different agents. So to make this scheme, a lot of factors were taken into consideration, like characteristic of what is pathological process is involved in endometriosis, what is pathophysiology of it, and this is so the two things are important. First, that endometriosis develops from this mesenchymal stem cells, and what roots they are formed. The second is that depending on peculiarity of epigenetic factors, there can be different forms of this endometriosis. This was with endometriosis first stage, so-called, of form, second, third, fourth form, or it is even more, can be hyperplastic process of endometrium, including as a marginal effect, a carcinoma of the uterus. And when later we took the systemic approach to myoma, I'm not mention all details, but then we found that it's possible there's sufficient ground to suggest that in each uteri there is a layer between myometrium and endometrium in the wall of the uteri, and this is the main source of these mesenchymal stem cells. And in respective of some particular mutation or action of provocative agents, which are written here in Russian, it can be it can give rise these cells either to myoma or they can give rise to adenomyosis or endometriosis. And this is a rather interesting thing and gives you clue for diagnosis and for prevention these diseases. And just if not so much is left, I should coming to the end. I was also happy enough to see that somewhere in 2013, the European Association for Predictive, Personalized and Preventive Medicine was formed, and they started with editing the book, and the name of this book is given both in Russian and in English. And its idea is to make a pathway of development of predictive medicine. So first of all, they really come back to the predictive medicine again at a new level. And second, they suggested this pathway. It's a rather complicated pathway. I will not speak much about them, but only I should say that it is suggested for maybe 40 years. So within 40 years, when they think that we can implement all these ideas, all the methods, all these approaches in medicine. And here, as you see, this very famous face, January 26, 2015, there was the Obama, probably after he was suggested by Francis Collins, approved the so-called the program of precision medicine. And they started very quickly to move this direction. I should also pay attention that even though in 2013, Francis Collins, which I mean, already created in the United States Patient-Centered Outcome Research Institute, which actually the main idea was to compare all clinical peculiarity of the patients with peculiarity of its genome, just to find out what was the reason of these differences. And as usual, Americans are moving very quickly. They not only want to make, they want to spread it to everybody, and everybody who has a device like you have, how you call it? Planchette? Tablet. Okay, let it be a tablet. That if you have tablet, you can participate in this study yourself because you can address to genome, to your genome, to inform your genome will be saved somewhere, and you inform what disease you have or what features, anything about you, you can tell to the bank where these data are collected. We are very coming closely to conclusion. And these are important, so I will translate a bit more in detail. So, deciphering of human genome explain the transition of all modern medicine on molecular or genetic genomic level. The other important second conclusion is whatever biochemical reaction you are doing, whatever laboratory analysis, including this one sophisticated and suggested here, which you discussed, they only reflect what's happening at that moment in organism. They not reflect the long-standing program. They change every day, Every day you change your diet, every day you change your breathing, every day you change your acquaintance, and so on. But there are one system which does not change. And this is the genome which you received from your parents, which is almost have no change throughout the life. So analysis of genome gives you absolute information, and you should do it only once in life. You don't need to do it every day. Predictive personalized preventive medicine is a natural product of invasion of achievements in genetic in medicine during the last 15 years. It has already had a long way from genomic medicine through translational medicine to precise medicine. Genetic testings today can help to identify the group of diseases which are dangerous to this or other particular man. But it can't say for sure that you get these diseases. This is we can't foresee it. But we can guarantee that you according to the gene field, you are prone to that or another diseases. It's important that owing to, at the present, it's rather important to stress that before we studied the anatomy, when we looked for genes, when you look for mutation, this is what we studied, the anatomy of the genome. Just as if it is some organ and we are looking for sulcus, for foramina, and so on. This is just a skeleton of the life. But idea is to look for how it works, to look for dynamic genome, so-called. And this can be understood only through the way of using systemic biology of the genome. The sequence of new generation sequence, and the creation of unified genetic and clinical databases. This is the highway to the predictive medicine of the future. Evolution through predictive medicine to integrative medicine, and later to the precise medicine. I have finished.